Hello, everybody, and welcome to Creation Myths. StreamYard is telling me we are live. I'm going to wait and make sure that I can see and hear us on YouTube. It looks good. In the chat, if you're listening, if you're watching, please let me know if there's any sound or video issues. I will see it in the chat, so just let us know. So hi, everybody. Welcome back. It's been a while since we did something like this, but we have a really fun show to do tonight. Because we are going to be talking about a recent piece from Creation Ministries International called 40 Failed Predictions of Evolution. I'll be pulling up that article as we talk about it because we're going to be jumping through it up and down. But basically, it's 40 supposedly failed predictions of evolution, and we're going to talk about why they're all nonsense. We're going to get through as many of them as we can. Before we get into anything, the first thing we need to do is introduce my fantastic panel for tonight. So I'm just kind of going to go around in the order you are on screen. So Mr. Wilford, tell us about yourself. What do you do? What's your specialty? What are you going to talk about tonight?
Hi, I'm Mr. Wilford. I'm a geology undergrad at the University of, well, maybe that's not the best thing to say, but I'm here to talk about the geology claims in this article, at least as many as time permits.
All right, Dr. Zach Hancock, who are you and why are you here?
Hello, I'm Zach. I'm a postdoc at the University of Michigan. I study spatial population genetics, and I'm here to talk about the genetics part.
Jordan, from Reason to Doubt. Wait, Zach, you have a channel. Tell us about it.
I do have a channel. The handle is TalkPopGen, and I do a little bit of like pseudoscience debunking, some evolution education, and a little bit of like history of science.
And by the way, everyone's channels here are linked down below for all the guests. So they're down there. Actually, Mr. Wilford is a late join, but I will put your blog down below after the show so everybody can find that. Jordan, tell us about yourself.
I am a nuclear engineer. Being an engineer, I'm an expert in all topics, but I'll be confining myself mostly to just the physics stuff because I am a nuclear engineer, and I heard a vicious rumor that they're going to talk about radiation. But if you want to hear me talk more with a bow tie, then you can go to my channel. We talk skepticism. That's what we do. It's Reason to Doubt.
It's great. Everybody should check it out. Grayson, I think this is your first time here, so welcome. Tell us about yourself.
Thanks. Yeah, my name is Grayson. I've got the YouTube channel Based Theory, where I do debates on all kinds of pseudoscience, from young Earth creationism to flat Earth to climate change denialists. Pretty much open to any pseudoscience. I love science of all kind. I'm kind of a jack of a lot of different trades and a master of none. I do videos on topics from, physics to evolution, biology, everything in between. So I'm just going to be picking up the slack from some of the points that I think are some of the most hilarious and interesting to touch on tonight.
Awesome. And Jackson, who are you?
Hello, my name is Jackson. I am just as of right now, graduated with my degree in field and organismal biology.
Can't even remember what the degree is.
I did. I forgot.
Well, he's not an engineer. How do you expect him to remember stuff?
It's true. It's true. Biology is not real. Just like species. Species aren't real. Anyway.
Correct.
And so my not really expertise, I guess, is in zoology. And so my channel predominantly, which is also just Jackson Wheat, is about zoology. Right now I'm doing a long series going backwards through the history of life. And we're in the ray fin fish now, so that's cool.
All right. Well, thank you, everybody. Before we get going, just a very brief kind of little background for what we're doing here. So this article, which I'm going to pull up in just a minute, is called 40 Failed Predictions of Evolution. And the thing here that the author tries to do is basically say, because we have these failed predictions for evolutionary theory, we have to reject evolutionary theory. That's kind of the overarching kind of thesis of this article. And the reason for this is how the scientific method works. So for those of you watching who are not familiar with how scientific method in general, this is like the intro level that like I teach my undergrads, how the scientific method works in general, is that at the stage where you make a hypothesis, That hypothesis is your proposed explanation for something, right? You're explaining an observation, you're explaining a mechanism, you have a proposed reason how and why something happens the way it does. And to test that proposed explanation, you make predictions. And these are things that must be the case if your hypothesis is true. And like, that's the logical flow of it. If my hypothesis is true, then X, Y, and Z has to happen under blah, blah, blah conditions, whatever, right? And then you go out and you collect data, you do experiments, you make observations to test your predictions and see if they're accurate or not. Now, if your prediction turns out to be accurate, great, you have supported your hypothesis. We never prove anything in biology. You can prove stuff in philosophy, you can prove stuff in math. You can't prove stuff in biology, but you can support your hypothesis by having lots of accurate predictions. If your prediction doesn't match your hypothesis, then that can mean your hypothesis is wrong. It could also mean that your hypothesis is incomplete, right? Maybe there's information you don't have. It could also mean you like messed up the pH when you did your experiment and you have to like fix your experimental procedure. So there's lots of reasons your prediction can be wrong. But the key thing is that it's part of this logical flow for testing your proposed explanations. And this is the foundation of how science is done. We propose an idea, and then we make predictions and test those predictions. And I'm going to give everybody a brief example of where a specific prediction was incorrect because we had incomplete knowledge when it was made. And then the underlying model was basically okay, but incomplete due to our lack of knowledge at the time. And the example I'll give you is basic Mendelian genetics, where back in the 1860s, Gregor Mendel said that every locus in the genome, every allele or every gene in your genome, he didn't have the word gene, he said heritable factor, he said all these different parts of a genome are going to do this thing called independent assortment. They're going to assort independently during meiosis. And his stuff with the pea plants turned out that all the things he used when he was doing his experiments assorted independently. We now know that that's wrong because like 40 years later, we realized that chromosomes carry genes. And so things on the same chromosome don't assort independently. So that specific aspect that Mendel proposed, that specific prediction from Mendelian genetics was wrong because he didn't know that like chromosomes were a thing in genetics. That doesn't mean Mendelian genetics is wrong, right? You can still find Mendelian traits that operate the way he said they should operate. It's just that he had incomplete information. So that's an example of where we have predictions that are wrong, but it's not because the underlying idea is wrong. It's because we have incomplete information at the time the prediction is made. That's a common thing in biology because what we're always doing is working towards more and more precise hypotheses to explain whatever it is we're trying to explain. A lot of what we're going to see in this article, as you'll see, is basically decades-old statements that are sometimes based on incomplete information. Sometimes they are just kind of out-of-context quotes that aren't really predictions in the formal sense. We're going to see a bunch of that kind of stuff in here. So I want to make sure we're all on the same page in terms of kind of where predictions fit in to the formal structure of doing science. And I want everyone to keep that in mind as we look through this article, because that is not a lot of what is talked about in this article. It's not predictions in that formal sense.
You can't trust science. It changes all the time.
All the time. All the time.
I also would just like to point out that very, very few of the so-called failed predictions in this article have anything to do with the theory of evolution.
Yeah.
Almost none of them, in fact.
Almost none of them. So I'm going to pull up this article for us, and what we're going to do is we're just going to go around the horn, just round robin style, and everyone's just going to pick one that they like, and we're going to talk and just going to nail it for just a couple of minutes. We're going to go as fast as we can, cover as many as we can. I don't know how many we're going to get through because there's 40 of them, but we're just going to get through as many as we can. So I am now going to pull up the article right here, 40 failed predictions of evolution. And Mr. Wilford, you're first in line there, right below me. So I'm going to start with you. Tell me the number of the prediction I should scroll to, and tell us why it's nonsense.
Scroll to number 7.
Let's go down to number 7. All right. Let's talk about paraconformities.
Paraconformities. So these are, in the actual geologic literature, there's a lot of debate over these. So we have a specific term for them, or a specific meaning of paraconformity, which means you've got two layers with no angular discordance, where there's evidence based on the fossils or radiometric dating or something that there is a time gap there, but there's not much evidence of any sort of erosion surface, or the erosion surface might be there, but it's pretty cryptic. What does that mean? It means that the layers are angularly equivalent, and there's not any topographic relief. Now, young Earth creationists only go by the second one. Like the Roth 2009 article that they cite, he will refer to angular unconformities, where the bottom layers have clearly been tilted and sheared off, but there's not a whole lot of topographic relief on the erosion surface. He'll call that a paraconformity. Of course, to a geologist, you can tell that the underlying layers were tilted and eroded. It's just whatever caused the erosion didn't make a bunch of relief. So we wouldn't actually call that a paraconformity. So they're basically complaining that if you've got a long time gap, you have to have a lot of topographic relief. And what's funny is that my most recent course was in sequence stratigraphy, and talking about unconformities, or another word we'll use for them is sequence boundaries. There are a bunch of processes we were taught about that do actually make fairly low relief surfaces that can be very planar on wide scales. One of the more well-known ones is called transgressive ravinement or wave ravinement. This is what happens when you have an erosion surface. It might have hills and valleys. It might have fluvial channels on it. But if the sea doesn't rise fast enough when it's depositing the layers behind the surf zone, that surf zone, those pounding waves will just knife off any erosion surface. And it'll get rid of paleosols, it'll get rid of weathering zones and granite, and you'll be left with a fairly conformable contact where there's not much evidence of topographic relief. That's just one. Paraconformities have also been linked to deep water bottom currents. Specifically, the Marshall paraconformity is off New Zealand is linked to the Antarctic circumpolar current system that opened up sometime in the Cenozoic. And then other paraconformities have been linked to widespread wind erosion called wind deflation, which can make erosion surfaces that have next to no relief on them. And there's other processes that can be talked about too. I mean, you could go into whether or not the old Davisinian style of peneplanation, does that ever happen in nature? We think it's rare, but it sometimes still does, even though the cyclic concept is wrong. There's different ideas about the lateral beveling of rivers, depending on how fast sea level falls and to what extent. But basically, they've got this simplified version of what they think erosion does, and it just doesn't correspond with what we know from sequence stratigraphy and sedimentology.
Thank you, sir. I don't know what a lot of those words mean, but that sounds comprehensive because I'm just a stupid biologist. I don't understand that stuff. But
you know how I feel when you talk about biology.
There we go. We're all in the same boat, except for Jordan. He knows everything. All right, Zach, what do you want to talk about?
Yeah. So speaking of biology, let's go to number 32.
Number 32.
Yeah. Sorry, we're gonna skip down pretty far.
Oh no, that's fine. Let's do it. Which one is 32? Oh
30.
My goodness. All right, let's hear it.
Yeah. So okay. So there's a lot in this one. The prediction is a low mutation rate in complex organisms. Okay, that's the prediction. They're using this to kind of set up a bunch of other things that have to do with like genetic entropy, and this panel can obviously speak a great deal on the idea of genetic entropy, but I actually don't want to go that way with this, the way I want to answer this. I want to stick on the prediction itself, because this prediction is actually the exact opposite of what evolutionary theory predicts. So evolutionary genetics has this prediction that selection should act to reduce the mutation rate, right? Because in general, mutations are deleterious. So selection should reduce that mutation rate.
I'm just going to stop you right there. Genetic entropy confirmed. You heard it from Zach. That's it. Game over. All right, keep going.
We can do the genetic, and I just feel like that the whole thing can turn into a night of genetic entropy, and I'm trying to avoid that. Prediction: low mutation rate in complex organisms. If selection is acting to reduce the mutation rate, because again, mutations are generally deleterious when they occur, then we can make a prediction. because based on the efficiency of selection. So what is a complex organism? Well, complex organisms tend to be eukaryotes, they tend to be multicellular, they tend to be large bodied, and all of these things, as you're getting bigger and bigger organisms with more complex tissue types and like social structures, this means that their population sizes get smaller because these organisms need more space to just live and survive, right? Whereas very simple organisms tend to be very small, like prokaryotes, and they tend to be widespread and like in gigantic population sizes. So if the prediction or if the idea about like mutation rate evolution should be driven by the efficiency of selection, then the prediction from evolutionary genetics is actually that the large-bodied complex organisms should have the highest mutation rate because the efficiency of selection is weaker on them than it is in prokaryotes, right? And in fact, we can make a very specific quantitative prediction here that the mutation rate should scale to the inverse of the population size. So if you take a ratio of the mutation rate over the effective population size, when that value is equal to 1, we call this the drift barrier. And that's basically as efficient as polymerase or mismatch repair enzymes, as efficient as they can possibly be, is when that ratio is equal to 1. And so that is a very clear prediction. So prokaryotes, for example, have effective sizes between like 1e to the 10, 1e to the 11, like really, really large population sizes. What are their mutation rates? They are the inverse of that. 1e to the negative 10, 1e to the negative 11, exactly where we would expect those mutation rates to fall given their effective population size. So not only is this prediction just like backwards of what we would actually make, but it's actually a fulfilled prediction of evolution straight from mathematical population genetics. That's where I wanted to go with that one. I know there's like a genetic entropy component of this too, but like that's the main thing I wanted to go there.
But we're not going to do that. That was perfect. Boom, roasted. Jackson, you're up. What number are we going to?
14, please.
14.
Since we're doing this one at a time.
Number 14. Here we go. The fossil record. Yes. Hit
Yeah. So the prediction here is that, according to evolution, we should show diversity first and then disparity afterwards. So to define those terms, basically diversity is the spread of variations within a particular clade, while diversity is the spread of variations between two particular clades. So think, for instance, of like diversity would be like diversity within Arthropoda versus like the disparity between like nematodes versus arthropods. So that's the difference between those. Okay, so it says that the fossil record should show diversity first, developing into disparity, but the opposite is true. First and foremost, this is just wrong, first of all. But second of all, there's no model of evolution that necessitates the fossil record show this. There's no model of evolution, of any mechanism of evolution that has any bearing on the fossil record, really. We're just lucky we have the fossil record. It's really nice. But anyway, how is this wrong? So for one thing, this idea was debunked way back in 2005 by Derek Briggs and Richard Forty in their 2005 paper, Wonderful Strife, Systematic Stem Groups and the Phylogenetic Signal of the Cambrian Radiation. So this is playing on the idea that Stephen Jay Gould had back in the 1980s, which was common for the time, that the Cambrian represents this period of unparalleled disparity among organisms. So basically, you had like an inverted Christmas tree instead of what we look at today, or instead of how we understand the Cambrian today, which is our normal cone. And basically, what Briggs and Forty did was they showed mathematically that if you look at the disparity and diversity within clades during the Cambrian explosion, you do actually get your normal cone. You have low diversity, which then increases. But actually, the diversity of morphology that we have today in clades like arthropods is actually fairly similar to that of the Cambrian. So it's not an inverted, it's not a Christmas tree. It is just a normal cone. I think I misspoke earlier when I said that, but it's not a Christmas tree. It's not a triangle going, with the wide base at the bottom. But also, we do find diversity preceding disparity, and some examples. So we find that stem lineages precede crown lineages, which we should expect if evolution's true. So, for example, stem eumetazoans, like the rangiomorphs, those weird little frondy guys who appear in the late Ediacaran, in the early Ediacaran, that includes Charnia. So those appear before the crown eumetazoan, so before like jellyfish and bilaterians. Stem cnidarians like Hautia appear before crown cnidarians. Stem bilaterians like Dickinsonia and Icaria appear before crown bilaterians. Stem tetrapods, moving way forward in time, like Canichthes appear before the crown tetrapods, like amphibians. Stem mammals like Dimetrodon and Gorgonops appear before crown mammals. Stem birds appear before crown birds, etc. Over and over and over throughout the fossil record, this is just incorrect. So it's just wrong. They're literally just wrong.
I think that sums it up nicely.
That
Thank
could
you.
be the entire panel. We
It's
just
just-
say they're just wrong, and then
Everything. Just do the My Cousin Vinny and call it good.
everything they just said.
Yep. There you go. Grayson, hit us.
Yeah. So I was about to say, it seems like we've stumbled upon a theme here tonight. Let's go to number 5. And we
Number
can talk
5.
about everybody's favorite new measuring device to test the theory of evolution, the
There you go.
James Webb Space Telescope. So right off the bat, obviously has nothing to do with evolution, but I wanted to touch on this one first because I've seen so much misinformation out there about the James Webb Space Telescope and what it does and doesn't show. And I just wanted to what they've latched onto specifically in their criticism of evolution's predictions for what the James Webb Telescope should apparently see. They've latched on to that the prediction should be that there's no spiral galaxies and there should be no heavy elements in these early galaxies that the James Webb Space Telescope has seen. So first of all, I just want to say the whole, the James Webb is seeing impossible galaxies headlines that have been floating around the internet are pretty much totally off basis. There's no impossible galaxies that the James Webb has seen. It basically, the very farthest galaxies are measured by a Z-score, which it quantifies the amount of redshift. So the very farthest galaxy that is a spiral shape, like a bar spiral, the same type as the Milky Way, is around a Z-score of 3. So that's still pretty far. Like most of the other ones, and Z-score of 3 is a huge outlier, by the way. The other farthest ones are maybe Z-scores of 2s, but most of them, the amount of spiral galaxies goes down precipitously, and then you don't find any at Z-scores higher than 3. So already their claim that we're seeing all these spiral galaxies in the very earliest stages of the universe is just not correct. In fact, when we talk about the really far galaxies, like the earliest ones that the James Webb has been seeing, like Z-scores higher than 10, they don't look anything like spiral galaxies. In fact, I have what one of the astronomers that works on the James Webb described 2 of them, saying that one looked like a peanut and the other one looked like a fluffy ball. So
Was that the most recent article that came out? Because I think that one said they actually matched up fairly well with the predictions of the Big Bang model.
Yeah. So they do.
Yeah.
If you go and look at the pictures of what these very farthest galaxies actually look like, you can see what they look like to the James Webb. Just clearly visually, they do not look like spirals at all. They look like fuzzy little weird splotch objects. Their other claim that, we're surprised because there's all these heavy elements. Again, on the theme of that's just wrong. There's here is a quick study that's done over 100 astronomers working on the James Webb. We're working on this of the analyzing the spectroscopy for the galaxies with Z-scores higher than 10. So I should say that initially these galaxies were characterized by just a photonic Z-score. So that's kind of a rough estimate of what their redshift is. But then when there's more time, you can go back and you can do spectroscopic analysis on these galaxies, and you can get a much more precise Z-score for their redshift using spectroscopy and the Lyman break and everything. So there's a much smaller number of galaxies that have been fully characterized spectroscopically by the James Webb, but they looked at 4 of the very farthest galaxies that have really high Z scores over 10. And surprise, surprise, they're all metal poor. Like they all have very low amounts of heavy elements, which was directly contrary to what they claimed the James Webb was showing. And lastly, I just want to say, so they were wrong both on the spirals. They were wrong on the heavy elements. In fact, the actual James Webb results showed the exact opposite, that were in line with the Big Bang predictions. But it's even worse for the creationist, because within the Big Bang model, after recombination, you have all this like plasma recombine, like combining into like more stable, like non-ionized elements. And then once you have galaxies forming, they emit a lot of radiation that actually reionizes a lot of this gas. So that was a prediction of the Big Bang model, this reionization event. And then the James Webb can directly see that. Here's a quote from one of the scientists from Japan who was leading the study on this, that with Webb's data, we are seeing galaxies reionize the gas around them. So you can actually watch as these bubbles of reionized transparent gas grow around these galaxies as they decrease in their Z scores. So it's like directly confirming, like direct evidence of the era of reionization, which was a direct prediction of the Big Bang. So it's like, it's so much worse for the creationists than just the observations of the galaxy formation.
Beautiful. I didn't know any of that stuff. That's awesome. Thank you. Jordan, so you're going to just cover the rest because you know everything. So I'm just going to turn it over to you. And
everybody else can leave now.
There you go. All right. Hit us. What are we talking about?
Number 10. So 10. It's going to be unusual for me because usually I would stay away from geology.
I knew you were going to do this one. I love it. All yours.
But they made the mistake of putting something about radiation in their geology section. So that's why I'm here. So I'm sure you've all heard tons of times that there's carbon-14, and sometimes they say it's dinosaur bones or coal or oil, whatever it is, something that's very old. And the prediction, like they say there, is if it is in fact so many millions of years old or whatever, that there should be no carbon-14 left. And that is true to a certain extent. There should be no atmospheric carbon-14 left. So if you don't know, there's different kinds of carbon. Carbon-14 is the radioactive stuff that's in the air because of cosmic rays. And when you eat plants, when plants breathe and stuff, you get it in you, and you have about as much as the atmosphere has. And when you die, you stop breathing. No citation for that. And then it decays away over time. So after like 100,000 years, there won't be any left. However, this claim assumes that there's no other way that you could get carbon-14 in the sample when you test it. And there's lots of different ways. For starters, even if you measure nothing, if you just like run the machine with nothing in there, you're going to get a detection of carbon-14, because no lab, no matter how careful and precise they are, they can't all be engineers. So they don't all clean their. I'm going to stop using that joke. It's getting old. So there's always some like residual background or whatever. But additionally to that, so that'll get you like somewhere in the neighborhood of like a 50,000 year measurement, 50,000 year old, quote unquote, that much carbon-14 if you just measure it with nothing in there. So there's a small amount of error there, but we can do better than so. That alone would be enough to completely throw away this claim. Because usually when you see it, it's usually somewhere in the 40 to 50,000 year range when you see these things. But we can do better than that, because you can actually get carbon-14 from different sources, not just from the atmosphere. So there's a lot of different ways. You could have contamination by like bacteria or water or whatever that would have some of that atmospheric carbon in it, but you can also get it in a kind of a similar way to how you get it in the atmosphere through radioactive decay. So that's what I'm going to talk about now that I've built it up. So basically, uranium is the source, or could be the source, for carbon-14 and coal specifically. The way this would work is your uranium decays, it spits out some neutrons. Those neutrons bounce around. They don't like being absorbed by carbon, and most coal is carbon, so it bounces around in there. When it hits nitrogen, which is in there in trace amounts, nitrogen will absorb neutrons sometimes, and then it'll turn into carbon-14. Same reaction that happens in the atmosphere, and then that carbon-14 can decay away. So the uranium, extremely long-lived. It's not going away. It's like millions or billions, depending on the isotope half-life. So like there's It's going to be there essentially forever. You can do some calculations to see how much what the age would be if you're getting all of your carbon-14 from embedded uranium. And if you just use because the amount of uranium and the stuff in the coal changes wildly across the world. But if you just take average numbers, then you get a age from the uranium of 51,000 years. That's how much carbon-14 could be produced in steady state in this uranium. So then if you add in the error we already had, the 50,000 years you'd get from just contamination or whatever, that gets you to an age of 45,000 years. And this is before we had any kind of statistical variation or anything, right? We're already at 45,000 years just from this. Now, some creationists have tried to refute this by doing some fancy math. Specifically, the guy they'll refer to, he wrote a paper in the very prestigious Creation Science Quarterly Journal, and he did a whole bunch of math. His name was Russell Rhoda, and he showed that you'd need 100,000 times more uranium than exists in order to get the carbon-14 you need. Unfortunately, he wrote his math extremely weird and unusual. I don't know if that was on purpose to throw people off or what, but it was very dumb the way he wrote it. But I managed to decipher what the hell he was talking about, and I figured out he was using what's known as a thin plate assumption. So if you're throwing like a beam of neutrons at a thin plate, you can ignore the thickness, right? And that simplifies the math. And he was doing that in the dumbest possible way. Unfortunately, coal seams are 2 to 50 feet thick. Now, thin is kind of an ambiguous term, but for the purposes of this formula, 2 feet is not a thin plate. Basically, what a thin plate means is that if a neutron comes in, if it doesn't interact, it disappears and goes away forever. So what his formula is saying, if a neutron touches carbon, it is dirty, it is unholy, and cannot be used for anything from then on. It is a tainted neutron. So anyways, that among many, many other reasons is why this is nonsense.
And just to piggyback off that real quick, because that was a great explanation with the coal. With the diamond thing specifically, I've only ever seen creationists cite exactly one study supposedly showing carbon-14 in diamonds. And to my understanding, in that study, they were literally using the diamonds as a control to test the baseline of their instruments of the standard measurement error of their instruments, and using a material that was known not to contain carbon-14. And that's the only time I've looked on Creation's website for any other study showing C14 being present in diamonds, and that's the only one I've ever seen cited.
Yeah, I'll second that. It's a basically a calibration study. It's a hey, here's our background level kind of thing. It's it's bad. All right, that was great and thorough, and also I understood it, which is awesome when we're talking about something that's not biology. So I am going to, with apologies to Zach, I don't know if this one was on your list, but this one immediately jumped out to me as my favorite. I'm going to jump down to number 28. So number 28 is the prediction that there must be lots of junk DNA, and as we all know from ENCODE, there is no junk DNA anymore because ENCODE published that one paper in 2012, and now there's no more junk DNA. And so we know that was wrong. Oh my goodness. So let me just start with this one line here. First of all, right here, the ENCODE project found at least 80% of human DNA is functional. That's just a lie. There's no other word for that. That's not what ENCODE found. What ENCODE documented was in their original 2012 study was approximately 80% of the human genome is involved in some biochemical activity other than replication, because 100% of the genome replicates. So by this standard, you could just say everything is functional because it all does some biochemistry. It all replicates. But other than that, some protein binding, some transcription, some biochemical activity is present for 80% of the human genome. Even in the 2012 paper, ENCODE said, we're documenting activity. This is a good place to start looking for function. It doesn't mean specifically that it is definitely functional. Like, they are clear about that right off the bat. The press releases, which is what the author here cites, if you click on those links that are embedded there, he goes to the popular level writing about ENCODE rather than the actual ENCODE publications. But beyond that, you go to the 2014 ENCODE paper that creationists never cite. They are much more circumspect about their conclusions and document, for example, how, yeah, there's a lot of transcription, but some obscene amount of there's 75% of the genome does transcription, but of that 75%, something like 30% of it, the level of transcripts is fewer than 10 per cell, which means it's not doing anything. It's clearly just spurious transcription from, pseudogenes or transposable elements that still have some residual transcription activity, right? And ENCODE is clear about this, that they say, it's the case that if something is transcribed at that low level and the sequence is not under purifying selection, it's probably not doing something important in the cell. It's probably not functional. That's not me saying that. That's ENCODE saying that in their 2014 paper. So this is just straight up misrepresentation, and bonus points ignores the later research by the same team he cites. Now, the actual research shows that probably somewhere between 80 and 90% of the human genome is actually junk. This is what we know in 2023. And I just want to pitch Dr. Larry Moran's or Lawrence Moran's book, recent book, What's in Your Genome, as an excellent summary of all of this stuff, that most of the human genome is in fact junk. This is complete nonsense, what is written here.
Yeah, if I can just jump in just really quickly. I did recently interview Larry Moran over his book. So if you want to see that interview, check out that interview on my channel. And your purifying selection point, right? If we look across the genome and we see that, and really only 5% of it actually shows evidence of purifying selection. We give 10% as kind of a conservative estimate, but only 5% of it actually has evidence for purifying selection. And so what's key here is that if we say that 80% of the genome is functional, we're stating that much of the 75% of it can be maintained in the absence of selection. That means that any mutation that occurs at any point in the genome is never deleterious. It can never harm you. That is what that is saying. If the genome can persist with function and yet have no selection purifying those deleterious mutations, then you're suggesting either no mutations occur or they're never deleterious. And that's obviously absurd. To the very first prediction, right, that there must be lots of junk DNA is also just kind of an absurd point. We know the prokaryotes, for example, have very little junk DNA. So evolution actually makes no prediction that there should be lots of junk DNA just across the tree of life. It's that the amount of junk DNA should scale with the efficiency of selection. And that is to say, it should scale with the inverse of the effective population size, right? So the smaller the population size, the more junk you're expected to see. And again, this is another really well confirmed prediction of evolution.
All right. Back to Mr. Wilford. We have gone around one time completely, and it only took us 40 minutes. Y'all, we are never going to finish these 40. So I'm going to add, here we go. So,
okay. So real quick, I want to go back to number 7, the paraconformities one, real quick first.
We just talked about how long this was taking.
No, it's because I missed one thing I meant to add.
All right. Quick.
Okay, so again, a lot of young Earth creationists think that there's no erosional processes that will ever make these very low relief unconformities, but I want to point out a particular quote from young Earth creationist geologist John Whitmore. This was in the Journal of Creation in 2019. He said, while flat surfaces are uncommon in terrestrial settings, they are very common on the seafloor. So it is not surprising we see flat contacts between marine layers, which are most of the rocks in places like Grand Canyon, even from a conventional perspective. There is still missing time between many of these layers from a conventional view, but at least that is easier to explain in a marine setting. So, boom. Not all of them straw man this, but CMI decided to, I guess. Now, the other one I want to go to.
Give me the new one.
Real quick. Number 9. It's right there.
Number 9.
They say that sedimentary strata should have limited geographical extent due to the localized nature of depositional processes. That's not correct. The vast majority of especially marine strata in the geologic record are from the result of marine transgressions, where the ocean rides up onto the land, and regressions, where it falls back. And this causes environments like, for example, you'll get a strip of near-shore sedimentation that can be very laterally extensive one direction along the coastline, but is very narrow in its way going inland. But if the sea level rises, what is that narrow strip of sand going to do? It's going to start pushing inland, and it's going to spread out over a much wider area. Any modern environment, particularly in coastal settings, is only a snapshot of what the end product should be given changing sea levels. We actually have a name or a word for formations that are formed like this. They're diachronous. They're not the same age throughout. I don't know of many formations that are hundreds upon hundreds of kilometers wide in all directions, where any particular point is thought to be the same age throughout. Those mega sequences they're talking about, those are from transgressions and regressions of the ocean that spread limited environments over a wider area. So there's that. That's it.
That was awesome, and it was only 90 seconds, which, way to go.
You're welcome.
Yeah.
All right. Zach,
All right. Let's
we
do
going?
30, and I'm going to be really quick here as well.
30. We're going to 30. Where's 30? After 29. There's 30.
All right.
Oh, my goodness. This one.
Okay, so the prediction, the first living cell must have been quite simple. I mean, that's okay. But then they go on to state that researchers expected the cell to only have 20 genes, but now, and this is the key point, the proposed last universal common ancestor is getting increasingly complex. It now supposedly has over 400 genes. So this is a really critical point to understand about coalescence and what the last universal common ancestor actually represents. The last universal common ancestor is not the first cell. It is not the origin of all living things. It is just the last coalescent ancestor of all living things. That individual lived in a population of individuals. In fact, there could have been thousands of other species around at that time, bunches of other kinds of like proto-life at that time that just went extinct and didn't leave any genes behind. So it is the coalescent ancestor of all life, but it is not the only individual. Hence, this individual can be as complex like as you need it to be because it doesn't represent the first cell. There were many like proto-cells, pre-life forms that lived long before it. It does not represent the original thing. And this is true like anytime they start talking about things like coalescence, when you think about coalescence in human populations or coalescence in any other populations, whatever that coalescent ancestor is, whenever they live, that doesn't represent when that species originated. That just represents when the last, or in this case, the most recent common ancestor of that population lived.
Yeah, it's pretty sneaky of them to say, you know, last universal common ancestor, and then in the very next line say the minimum viable cell and conflate those two things. And totally ignore all protocells and seem to imply that genes and proteins were already necessary at the origin of life, which is like completely nonsensical. So
you say sneaky, I say a lie.
No, I mean, that's a bait and switch. That's just a straight up, it's taking this thing and saying it means the same thing as this thing when everyone knows those two things. And by everyone, I mean the author, John Batten, knows that those two things don't mean the same thing. He's just lying.
But their readers don't know that, right? They're betting on their readers not knowing that there's this bait and switch going on.
Nobody has more disdain for their audience than creationists.
Oh, yes. And I'm going to take a moment here as the host to just take a moment of privilege to point out that no young earth creationist has less respect for their audience than my bestie, Dr. Nathaniel Jeanson.
True.
Nobody.
Who they do cite in this, I hope we're going to get to that prediction because they cite Nathaniel Jeanson as having a successful prediction.
Oh, the stupid
boy
bird thing. Yeah, well, all right. Jackson, you're up.
15, please.
Nice.
Take it away.
Okay, so the prediction is that there should be, finally graduated organic chains of transitional fossils. This, harkens back to dear old Daddy Darwin, back in Origin of Species. And he wrote back then, I think it was chapter 6, I believe, was the difficulties on the theory, in which he talks about like that, the organs of extreme perfection, like the eye and the lack of transitional fossils, things like that. and he says, oh, these are major problems for my theory. It's been almost 200 years since then, and so we've made quite a bit of progress in this area of finally graduated organic chains. We were talking about before the show started things like Australopithecines, Tiktaalik, things like that. And within those, you have we have genera and leading to other genera. So we have I think a dozen different species of Australopithecus, and even prior to that, we have Sahelanthropus, Auroran, Ardipithecus, and we have Australopithecus. And then within that, we have multiple genera because Australopithecus is a mess. So you have Paranthropus, Kenyanthropus. Then Homo emerges from within that. And then we have Homo rudolfensis, Homo habilis, then Homo erectus, and we have actual a major a transition within Homo erectus, which is in terms of cranial capacity. So Australopithecines, like other modern apes, have a cranial capacity which is about a third that of extant humans. But in Homo erectus, you see it going from about a third up to almost what we have today. So it's quite a transition within one species, which should probably be multiple species. And there have been attempts to split that, like Homo georgicus, Homo ergaster, things like that. So just wrong. And then of course that leads to Homo sapiens. And then we have, we actually differentiate modern Homo sapiens from archaic Homo sapiens. So the first appearance of Homo sapiens in the fossil record around 300,000 years ago, they actually look a bit different than we do today. So that's just wrong. Of course, I love phytoplankton. I'm a botany TA, so I love talking about phytoplankton. Phytoplankton, some of them are little guys, unicellular eukaryotes that have essentially glass shells. So like radiolarians, diatoms, foraminifera. And these are deposited on the seafloor in absolutely massive numbers. And so when these turn into fossil-bearing layers millions of years on, you actually can see species to species transitions between foraminifera, diatoms, etc. And that's why they're used as index fossils. So they correlate, each species correlates with a very particular slice of time, and then they transition to another species because the environmental conditions change, and then they transition to another species because environmental conditions change again, etc. And in addition to that, we have ammonites, conodonts, which are primitive vertebrates. Among dinosaurs, we even have chasmosaurines. We actually have species-to-species transitions within the chasmosaurine lineage here in the U.S. So like Pachyrhinosaurus, Enosaurus, these are all part of a single anagenetic line, which is kind of crazy. Thalassocnus, which is the aquatic sloth from South America and Central America. Myotragus, which is a weird goat from the Balearic Islands. Look him up if you haven't heard of him, Myotragus. Hyopsodus was actually pulled by Philip Gingrich when he was arguing against punctuated equilibrium back in the 1980s, because you can see, again, species-to-species transitions within the Green River Formation. Paranthropus, I mentioned that one earlier, that also has evidence of microevolution within Paranthropus. Suffice to say, even though, yes, we have all these lineages, all sorts of different things, Eusnopteron, Panderichthyes, Tiktaalik, Parmastega, Acanthostega, Ichthyostega, all throughout the fossil record. Even if we had none of these, none whatsoever, there's a famous quote that gets mined by creationists from Richard Dawkins in Greatest Show on Earth, and it's, even if we didn't have any fossils at all, evolution would still be true on genetics. And that's true. We don't need any fossils, but again, it's really cool that we have them, and even still, this is just wrong. We have transitional forms in all these different groups.
And if I could just elaborate on one of those, because they claim, transitions between major body plans are never found. And like you mentioned Tiktaalik, but just to highlight that, how many fish can y'all think of that have lungs, a neck, and wrist bones? Like, that seems like a pretty a transition between some pretty major body plans there, from aquatic to terrestrial.
But have you considered amphibious assault vehicles?
Right, and they typically mean, when they say major body plans, they're correlating that with the phyla, which again is all taxonomic ranks are arbitrary. Yeah. So they're stealing from Stephen Meyer there, and I've done lots of stuff. Go watch the Professor Day video that I co-wrote on Stephen Meyer and Gunter Beckley. It'll answer all that. But yeah, even in those cases, we have STEM like arthropods, for instance, who don't have the full modern arthropod body plan, but acquire it stepwise, like Anomalocaris, Fushi, and Huia. I'm trying to think.
Would Proto-Articulata be an example of like even before the Cambrian back in the?
Well, that's like STEM bilaterians. Yeah, they don't even have they have like glide symmetry. They don't even have like actual bilateral symmetry. Yeah.
Okay, I'm gonna cut us off because we have a Jackson. You could literally do this for an hour.
Yeah.
Just go through all the different things. I think we have made the point. So I am going to kick it down to Grayson. What is your second pick, sir?
Yeah, I think that we could continue listing transitions for like the next several days. To be
just
honest,
go around the panel, just never stop doing the transitions.
For the sake of time, that would be a really fun stream. Let's go to number 18.
Number 18, right here. Here we go.
Yeah, this is a favorite little hobby horse of
Classic.
mine. Yeah, just because my degree is in biochemistry, just like when creationists claim, oh, what about the soft tissues in dinosaurs that Mary Schweitzer found, and oh my god, it drives me up the wall with that one, because it's like, if you actually understood like biochemically what Mary Schweitzer is looking at in these fossils, you would understand that there's no way that like the collagen fragments that she's describing could be only 6,000 years old. Like the collagen is so broken apart and cross-linked, which is very crucial, because every time I hear creationists bring up this example, they're like, oh, but collagen in an aqueous solution has this decay rate. And I'm like, oh, really? Like, what's that decay rate when you introduce all of these linkages and you introduce a lot of iron in the environment, which is inherently stabilizing? Like, they don't calculate how much those stabilizing factors will increase that decay rate and make it so that it's perfectly plausible for those collagen fragments to be lasting in dinosaur fossils. And especially, it should be noted that Mary Schweitzer's procedure literally involved demineralization of those bones. So she's literally just melting away like all of the minerals to just leave behind any non-mineral content. So it's
the
not like
of the bone matrix also acts like formaldehyde. That was uncovered by a recent study.
Yeah. So, I mean, there are all kinds of stabilizing factors that are just completely ignored here. And like, a very important thing to point out is that like Mary Schweitzer used to be a young Earth creationist, and then she actually studied the science, and then she made this discovery. And guess what? She's still not a young Earth creationist. If she actually thought that this was evidence to reify her previous position, you think that she would have said so, but she has explicitly come out and said, creationists, please stop using my results to try to push your agenda. Like, it doesn't show what you're trying to say, it shows. So I just wish, it's like a pet peeve of mine, creationists, please stop bringing up Mary Schweitzer. She doesn't appreciate it, and you're just dead wrong on the biochemistry.
And there's a really good book by Fazal Rana on this whole topic. It's Dinosaur Blood and the Age of the Earth. You guys should
could not recommend it enough. It's so good.
And for the record, lest anyone think we're just a whole bunch of biased evolutionists here, Dr. Rana is a noted old Earth creationist, and is a delight to talk to. He's great. Okay, so I just want to plug our friend Dr. Joel Duff, because he has a series on this exact thing called the Hemoglobin Challenge, and it's basically creationists stop misrepresenting this research challenge impossible series of just a parade of creationists misrepresenting this, like, we found blood in the fossils thing that always happens. So
I would like to usher a challenge as well. Like, I mean, because what she was finding were extremely stable forms of some of the most stable, like, organic molecules in the body. So, like, creationists, how about you find some like DNA samples from a dinosaur? I would take that.
And then we'd see, and then we'd sequence them, and it would be like a chicken, and we'd be like, boom. Right, it would nest. It would be the sister taxon to birds.
Schweitzer do something
and modern
similar? Like some of there was some material from some of the soft tissue material from a tyrannosaur, which was like comparatively most similar to like ostriches. Didn't that happen?
That would make sense. I would assume.
Yeah, again, it's like birds. Yeah.
Mm-hmm. Okay. Our next thing, we just did Grayson, so we are back up to Jordan.
Number 3.
Number 3.
Going back up to some physics.
All the way to the top.
The only thing that anyone should care about
here. Dark matter. Let's do it.
So, what they're saying there is
Let's do it quickly. Let's do it.
dark
quickly.
matter is this this mysterious thing. It's kind of a rescue device. You know, no one's ever been able to detect it. They're just making it up. Okay. And here's the thing. That's again a lie, because dark matter isn't hypothetical. It's not a theory. It is an observed fact. We know dark matter exists. We don't know everything about it, but its existence is not in question. And so there's a lot of different ways that we know it exists, but some interesting ones include so galaxy collisions are one of them. So when you have basically dark matter is matter, meaning it has mass, it's affected by gravity, it doesn't emit light, hence dark, and it doesn't interact with other matter very much, because if it did, we'd be able to detect it easily, right? That's all dark matter is. So one of the ways you can detect it is you can, first of all, just look at a galaxy. You can detect how much gravity is there through like gravitational lensing or things like that. But you can also like look at the amount of light you have and do some math with how big stars are, etc. And you get a mass estimate from that. You get a mass estimate that you can detect from the lensing. And guess what? Those two are very, very different. Well, it looks like there's a lot of matter there that isn't emitting light. Dark matter. You can also see it in collisions of galaxies. So if galaxy clusters hit each other, the gas, the regular matter, is going to slam into each other and generate a bunch of X-rays, right? And they're going to kind of chill out in the middle where all the X-rays are. But the dark matter, it doesn't interact. So it's just going to pass by like two ships in the night. So what you'll see is a glowy bit in the middle, bunch of X-rays, and then two globs of gravity matter on either side that doesn't emit light. That's dark matter right there. You can literally see it in a telescope. You can also see it by measuring the rotational speed of galaxies. I won't go into how they do that, but just trust me, they can. And if all the mass in the galaxies was coming from visible matter that emits light, then it would be going, the arms would be going a very different speed from the core, but they don't. There's a bunch of extra gravity there, and none of it is generating light. It's dark and it's matter. It's dark matter. So again, none of this is hypothetical. This isn't a question. We don't know what it is. We haven't been able to like find it in a lab, but that doesn't mean we don't know it exists. We just know it exists way out there. We haven't yet synthesized it here. That's all.
And if I could just highlight just one specific lie that they have here that I think is hilarious given like recent events. They say, now it seems as though a thorough application of Newtonian mechanics to galaxy star orbits explains them without dark matter, which like literally 2 weeks ago, a study just came out like observing like wide binary star systems and ruling out this modified Newtonian gravity alternative to dark matter. They ruled it out by like 5 sigma. So like it was like incredibly debunked. And like modified Newtonian gravity is like literally like it's like it's dying at the moment because of like I don't know. It's not, dark matter is pretty hard to find a workable alternative to. Them trying to present that, oh, we could just modify Newtonian gravity. No, it doesn't work. I dare anybody to try to explain the bullet cluster, which Jordan was talking about with the cluster colliding. Explain the bullet cluster without dark matter. I dare you.
One other thing about dark matter, Danny Faulkner, who's a young Earth creationist astronomer with Answers in Genesis, in like I think it was like 2018 or 2019, he wrote a piece begging his fellow young Earth creationists to stop denying dark matter exists. There is just too much observational evidence for it. So to me, that settles that question.
Just want to say I was fast.
You were. Thank you, Jordan. I'll just add from the chat that the study that Grayson just mentioned, it was 16 sigma. That is a high number. That is that is statistically significant. We can say
that is a stupid amount of sigma.
Okay. So that was number 3 from Jordan. Thank you. We are back to me, and I am going to bring it up to number 33, or I suppose down to number 33, because this one made my brain hurt when I read it. Zach, once again, I apologize because I suspect this might have been on your list too. So if you want to contribute once I'm done, this is the mutations are not random thing, which is based on a very recent study in, I think, Arabidopsis, which is a plant genetics model organism, where what creationists have taken this study to mean is that organisms can induce or cause or direct specific mutations to happen in response to environmental stimuli. That is nonsense. That is not what that study said. So I want to just clarify some language here, because the word random misleads people, I think, because you say random, you think like flipping a coin or rolling a die where every outcome is equally likely. But in the technical sense, that's not what random means. So a word that I like that I think is more precise is that mutations are probabilistic. That means that they occur along a probability distribution within your genome. There are certain regions that are hotspots that are more likely to experience mutations. There are certain regions that are cold spots that are less likely. There are certain types of mutations that are more or less likely than others. But which specific mutations that occur in your genome at any given time is probabilistic rather than deterministic. And that study that the creationists have latched onto, they just love it, doesn't say that mutations are not probabilistic. What it says is that the species in question can modulate their mutation rate in a region-specific manner within their genomes. There are certain parts of their genome that they can cause to have more or fewer mutations. This is the first time that have been documented in that species, but there's lots of things that modulate their mutation rate, because mutation rates are phenotypes and they're subject to selection. Sometimes you want more, sometimes you want less. But what all of these things have in common is that none of the species that do this direct specific mutations to happen. All they do is they sample more mutation space faster to see if they can find a beneficial mutation, sometimes in a region-specific manner and sometimes not. This idea that organisms direct specific mutations to occur in response to environmental stimuli was disproven, and I can say that word because it was conclusively disproven in the 1942 Luria-Delbruck fluctuation test, one of the all-time classic experiments. So this idea that creationists have come back to was literally like disproven. And again, we can say that formally, it was disproven. It was very simple experimental design, and it was disproven over 80 years ago. I just shake my head. Zach, do you have anything you want to contribute there? Because I know that you read that one and also got a headache.
Yeah, I'll just say that like the Arabidopsis study is grossly misunderstood by creationists, obviously, but it's also, I want to point out, probably not the last word on this particular thing. And this is like a weirdness in scientific.
Not even the first word anymore.
No, but there has been 4 or 5 papers that have come out since that paper was published, critiquing it basically. And the authors of that paper did try to make this sort of directed evolution or sort of directed adaptation argument. But there have been at least 4 papers that I can name off the top of my head that have come out, one attacking them methodologically, another one that was attacking them theoretically. And I think the most convincing is showing that actually their experimental design cannot distinguish between directed mutation and actually just purifying selection. Because what they basically found was that you don't get mutations as often in functional regions as you would expect. But like, that's what purifying selection does, is it removes mutations from functional regions. And so their experimental design was actually inadequate to be able to distinguish those two things. And so, yeah, so I think Dan makes a really important point that obviously mutations aren't random with respect to where they occur. They are random with respect to their fitness effects, right? That you don't get a good mutation just because you need a good mutation, right? Mutations happen and they don't know when, like, what an organism needs.
Alrighty. Thank you for that additional point about the dispute of that study. Mr. Wilford, what is your third pick, sir?
I'm torn between 2. You know what? Let's go to number 16. It's in paleontology, but it's technically geology.
All right, number 16. Oh, there it is.
Fossils should
Our
not
favorite.
cut across many strata representing millions of years. This is the polystrat fossils. You hear this all the time. Simplest answer I can give, Erica has a really good video on this about how the overwhelming majority of these things, in fact, all of them to my knowledge, are found in environments like floodplains or volcanic deposits or similar environments that have a very high rate of accommodation. So they build up sediments very quickly. Joggins, for example, up in Nova Scotia, it's a very well-known heritage site for the USGS that has a bunch of different units of polystrat trees. Well, they're not trees, they're like copsids, but you get the point. And this formation, it's pretty thick. It only represents 1 million years. The beds that bury those trees, they're not millions of years apart. They didn't build up under millions of years of accumulation. It wasn't millions of years between depositional events like these guys say. The whole formation is under 1 million years. You don't see polystrat fossils in like very deep pelagic sediments where oozes accumulate at a fraction of a millimeter per 1,000 years. You see them in these high accommodation environments. So there you go.
And there's also been like secondary growth that's been demonstrated. Like
Yeah, that too.
The polystrate tree or whatever has like started to be partially covered by sediment. You can literally see it like growing after that happens, which would never happen in like a global flood in less than a year. So
yeah.
Nice and quick. Zach,
Yes. I'm going to be quick as well. Let's go to number 19. And I know this is one that Jackson probably also wants to talk about, so feel free to add something at the end of it. I'm going to make a theoretical pop gen argument, and then you can take it from there. So this particular one is about stasis. It's about evolutionary stasis, that fossils of organisms found in geological layers, separated supposedly by many millions of years, should not be, similar. They should be changing through time. So that's their proposed prediction. At best, this is a critique of Darwinism. So this is a critique of gradualistic Darwinism. We have not been under a Darwinist paradigm since like 1890. Okay, like Darwinism is not a thing that we've been under for a long time. The modern synthesis does not predict this. Ever since the founding of theoretical population genetics and quantitative genetics, it's been clear that most evolution is stabilizing. There's a ton of both theoretical and empirical work that shows this. So directional evolution only occurs when the environment changes. And by environment, I mean both like the biotic and the biotic. So when organisms are exposed to something new, and they have to, sort of adapt to it. So this is both theory and empirical research shows this, and that this adaptation happens really, really quickly. So populations can rapidly climb to some local fitness peak. And then from there on out, you have stasis, where stabilizing selection is acting to try to keep them on top of that peak. So long as the environment is not rapidly changing all the time, or even like gradually changing, then an organism should actually find a phenotypic optimum pretty quickly, and stabilizing selection should be the norm. And in fact, like when Gould came out, when like Niles Eldredge and Stephen Jay Gould came out with their punctuated equilibrium, the founders of the modern synthesis and a lot of like population geneticists were like really unimpressed with the idea. So like they pointed out to the fact that like people like G.G. Simpson and like a lot of like R.A. Fisher, like a lot of these people already had these notions. Like they already had the idea that most evolution should be pretty stable. So like, and they rebutted that idea that like, bro, you're just not up on the literature. This is exactly what we expected to see. So again, stasis was not surprising at all, and that rapid shifts between these adaptive peaks actually matched theory going all the way back to R.A. Fisher's genetical theory of natural selection in literally 1930.
Before Jackson jumps in, I just want to say, who else thought that this was probably the case earlier than Gould and Eldredge? A guy named Charles Darwin, you may have heard of him, who wrote in the 5th edition of Origin of Species, he said, quote, "The periods during which species have undergone modification, though long as measured in years, have probably been short in comparison with the periods during which they retained the same form." 5th edition, Origin of Species, 1869.
Yeah.
Oh, snap. Yeah, I just did a video in which I absolutely demolished this idea of that that organisms are like living fossils. I know it's called the coelacanth's tail, because both, if you look at the genetics of coelacanths, of which there are 2 extant species, their substitution rate is not abnormally low. Like it's like they are occurring mutations and they're being fixed in the population at pretty normal rates for fish. And also, just You know what creationists will never do? They'll never do a morphological analysis because they will literally never compare the morphology of or extant latimeria against their closest relative in the fossil record, which is called Swinzia. They're zero, not one to date. They won't do it because they can't substantiate this. There are differences in the jaw morphology, in the skull, like the opercular bones, all this. It's different from their closest fossil relatives. It's so stupid. Go watch my video, The Coelacanth's Tail.
Go watch his video, everybody, after this video. First watch this video, then go
Yes.
watch his video, Coelacanth's Tail. Jackson, you can just stay on the mic. What number should I scroll to?
Oh, 13.
13.
Yeah.
13. No organism wholly soft can be preserved. Let's do it.
Again, this is. I don't think this is this is not a model. Not a prediction of any model of any mechanism of evolution. It's certainly not a prediction of natural selection or sexual selection, or literally anything. This was just as Dan said earlier, an incomplete understanding of fossilization in the fossil record. And so, yeah, in Darwin's time, when there were far fewer known fossils, the thinking was that only hard parts could be preserved. And as I mentioned earlier, with regard to like phytoplankton, they are hard organisms like that. They are really good at getting preserved. The things we have the most of, like, you go to the chalk formation, the Dover Mountains in Dover, are like phytoplankton because they
White
have they're
Cliffs of Dover
coccolithophores.
for everybody.
White Cliffs of Dover, there you go. Yeah, they're coccolithophores, which have again this tough exoskeleton. We do have soft-bodied organisms in the fossil record, but they're in very, very, very particular fossil formations. They're called Lagerstätten, and they only form under. I'm sure Mr. Wilfer can talk about this greater detail than I can, but basically, you don't have this sort of deposition in most environments, and most organisms get scavenged after they die. And so, even if you do have, even if bones are left at all, a lot of times you'll find teeth marks in them, or they'll be reworked by like worms, things like that or bacteria that burrow into it. So this just isn't a prediction of evolution. This was just a lack of understanding of the fossil record, given that Darwin lived 200 years ago.
Nice. Grayson, tell me where to go next.
All right, let's go to number 4. We did
Number
dark matter,
4.
so let's do dark energy next.
There it is.
Fun one. I personally love dark energy. It sounds weird, but I think it's one of the coolest mysteries out there. So I would say that it's probably a little bit more questionable than dark matter, for example. Like dark matter is pretty solid. There's still a lot of a lot of evidence for dark energy, though. So again, similar to what we saw with dark matter, there's multiple independent lines of evidence to support the need for something akin to dark energy. Again, we don't know what it is. There's multiple competing hypotheses for what dark energy would actually be, or be composed of, or be caused by. But the need for dark energy in our observations is pretty apparent from multiple independent lines of evidence. The only one that they bring up here is by looking at the type 1a supernova data and just saying, oh, you have to assume that the redshift is caused because they're accelerating away from us. Well, no, the reason that we know that redshift is because of the expansion of space-time is because there is no other physically plausible explanation for it. Like, I mean, you can look at tired light hypotheses. Those have been debunked because there's not a blurring effect that we would observe that would be necessary for light to be losing energy as it moves. There's also the idea that this could be some sort of cosmic dust that would be redshifting these things. But again, when we look at things like the cosmic microwave background, which there's tons of intersecting lines of evidence from the cosmic microwave background reifying dark energy, it's not possible that could be caused by cosmic dust. It would interfere with the spectrum, and it's literally the most perfect black body spectrum observed in nature, which would not be replicable by cosmic dust being the explanation. So there is no alternative physically plausible explanation. And notice that no creationist offers one that's viable either. They just try to say, well, maybe there's some new physics out there, which is like even worse than positing dark energy. So like at least dark energy has multiple independent lines of evidence substantiating it, not just from like type 1a supernova data, which is pretty clear. A Nobel Prize was given out for this discovery because again, it was not something that was expected. I mean, Einstein came up with his cosmological constant, but that's not exactly a prediction of dark energy. You could say that, well, actually, what they say here is that the prediction is that there's a lot of dark energy that exists in the universe, which is weird because dark energy was kind of a surprise in the 90s. But okay, if they want to say that the prediction is that there's a lot of dark energy, then our observational evidence absolutely reifies that prediction. Like I said, you can look at like there's these things called baryon acoustic oscillations in the cosmic microwave background. And when we observe those in some of like the smaller peaks, those match with predictions from dark energy models. We can look at the large scale structures of our universe and the various the voids and more dense regions that also matches with dark energy as was corresponding in the cosmic microwave background. We can, like, again, the supernova data is pretty clear. I mean, there are multiple converging lines of evidence supporting dark energy. So again, it seems like the prediction, rather than failed for this one, their prediction that there's a lot of dark energy in the universe is pretty reified. And it will probably continue to be so in the most latest Euclid telescope that was just launched into space and just started sending back images, I think 2 weeks ago. One of its sole purposes is to further understand dark energy. So we can look forward to that in the next couple years as creationists continue to be wrong.
Excellent. Thank you. I'm coughing a lot because I have bronchitis, so I'm just going to turn it right over to Jordan and go back to muting myself.
Number one. So this is talking about the cosmic microwave background, so they say the prediction is that the CMBR should be very lumpy, but it's extremely smooth. And so cosmologists invented inflation because of this. It's a rescue device. They constantly do this where they pretend like physicists are really worried about creationist models and like are inventing things to save themselves from it. Also, point of contention in the article they cite, he talks about how the COBE was fuzzy and ignores the additional decades of research since then. But anyway, so inflation was not invented for this purpose. The basic argument they're making here is that the early universe would need to have been lumpier than the CMB indicates it was in order for galaxies to form. And to rescue the model from there, you need inflation, this rapid expansion, which would magnify these tiny variations from quantum mechanics, quantum fluctuations, these tiny variations in matter distribution to allow galaxies to collapse. But that was a like side benefit of inflation. That was like something it solved along the way. The reasons it was invented was to solve problems of flatness. The universe is nearly flat. Flat in this context means lines behave like lines would expect instead of doing stupid things that you wouldn't expect. The horizon problem, the temperature of the galaxy, as far as we can see that way, and as far as we can see that way, are the same. But how would they know what temperature to be? Because they would never have been able to talk to each other. That's the horizon problem. And monopoles, where are they at? There aren't any that we can see, but there should be lots. Why not? Well, if the universe expanded a ton, then they would just be diluted away. Dilution is a solution. And so, is inflation true? Most people think so. There's a lot of good reasons to think so. But it wasn't invented as a rescue device for this one thing. It was invented to explain away a lot of stuff. You can have Big Bang without inflation. You just have to assume that the initial conditions were a certain way. But you can just remove that inflation, those assumptions, if you have inflation. So it makes your model simpler. It explains a lot of stuff. It explains stuff you didn't intend. And it's also testable. Something they say in their citation is that this is an unobservable thing. It's completely nonsense, but it is detectable because it would lead to primordial gravity waves. Probably heard of gravity waves. We have a detector with lasers going different ways, and they can detect neutron stars fighting. Well, you can do the same thing with inflation. They're just much smaller, so we can't detect them yet, but we should be able to detect them at some point, hopefully in my lifetime. And then I'm sure creationists will just accept the new science and close up shop, and that's what'll happen.
Another like successful prediction of inflation was that we should see adiabatic density perturbations in the cosmic microwave background. Then we saw exactly what was predicted by inflation with that, or at least evidence in line with that prediction. So there's yet more successful predictions from inflation.
I mean, if you want successful predictions, the fact that the cosmic microwave background existed at all was a prediction. And yeah, so they complained in their article, oh, well, not every single prediction was right on the temperature. Well, yeah, there was a range of predicted temperatures, but like, come on. Well, so this guy said it was 5, turned out it was 3. The whole thing is bullshit.
Famously, an XKCD comic is the cosmic background radiation. The science it works bitches comic is the cosmic background radiation. Thank you for that. That was excellent, and I didn't know that. I didn't know that stuff, so that's cool. I am going to pull number 29, which is related to the one I just did, which was 28, because it's all about junk DNA. And let me tell you, creationists are really bad about talking about junk DNA. So this one is specifically with pseudogenes. So pseudogenes are when you have a functional protein coding gene and then it mutates, like the promoter breaks or the start codon breaks, or there's a premature stop codon in the sequence somewhere. Basically, it no longer codes for a protein. So it's now a pseudogene rather than an actual gene. And the prediction that we're talking about here is the idea that pseudogenes don't have a function, which is true for the vast majority of pseudogenes. So what it says here in the article is that it turns out you can look at some pseudogenes and find a function, and that's true. That's true not just for pseudogenes, that's true for most classes of repetitive DNA, transposons, for endogenous retroviruses, for all of these classes of elements in our genome, you can find singular examples that have been selected to have a specific function. The vast majority of examples for each of those classes have no specific function associated with them. And specifically with pseudogenes, there's 2 ways we know that's the case. One is they are not experiencing purifying selection, right? The proposed mechanism for pseudogene function is that the non-coding RNAs transcribed from the now non-functional pseudogenes are going to regulate, and that's the key word, the creationists say they regulate expression and specifically translation of other similar genes, similar in terms of their mRNA sequence. What actually happens is that they can transiently interfere with those mRNAs and therefore prevent them from associating with a ribosome for a short period of time. But what we don't see for the vast majority of pseudogenes is, one, transcription at a level that's actually going to materially impact translation rates. And 2, something you would need for this to work, selection to maintain the sequence similarity with the functional genes they regulate. If you are going to regulate based on the fact that you match another sequence in the genome, you need to be under purifying selection to maintain that sequence similarity. And pseudogenes, very famously, are not subject to purifying selection, which means they are not functioning in the way creationists claim they do in terms of regulating gene expression. What actually happens with pseudogenes is sometimes the transcript from a pseudogene is similar enough to the mRNA for a functional gene that it interferes with translation of that mRNA. That does not mean it's been selected to regulate it. It just means it interferes with it by happenstance because they happen to be similar for at least part of their sequence. It doesn't mean that pseudogenes have widespread functions. That's just wrong. And he's just, again, keep using the word, lie, misrepresent, bait and switch, whatever you want to say. Like, he's full of it. He's being dishonest here. He's lying about what the research says here.
Yeah, he also says the very notion has discovered research to discover pseudogene functions. That's just BS. Like no one is like, oh, well, that's non-functional. So, 90% of the genome is non-functional. Therefore, I'm not going to research it. That's just a lie. Like, people every single day, like, there's a field of functional genomics. By the way, there is no field of non-functional genomics. Like, it's like there's only functional genomicists, and that's what they spend their day doing, trying to find function. So it's like no one is being deterred. Like, the research is still ongoing.
The if you want to know about, like, for example, those pseudogenes that have reattained function, look up what's called zombie genes. My favorite example is elephants have lots and lots and lots of different ways of mitigating cancer, and so they like refunctionalized a pseudogene that is like anti-cancer. So super cool stuff.
Nice. Thank you. Mr. Wilford, you're up. What number?
Go to number 12. So this is the claim that mixing of sediments due to the activity of marine organisms called bioturbation should be all throughout the Phanerozoic. This is actually observed. They make this claim that if it was there, then all sediments should be completely tossed up and there should be no bedding present. That's inaccurate. First of all, we do see a lot of layers all throughout the geologic record that do show intense bioturbation. And funnily enough, if you read the book that they cite for this, I actually got it on Kindle for this purpose, they actually recognize there are many horizons that do show intense bioturbation. And then that goes into their whole internal debate of this will help us decide where the flood is and isn't in the geologic record and so on. I myself have personally seen some layers here in Florida, like the Miami limestone, that are tossed up to high hell by bioturbation, particularly the oolitic facies. Really cool stuff to see. But yeah, lots of layers don't show much evidence of being tossed around by bioturbation, and there are a couple of reasons for this. One, a lot of thinly laminated mudstones form in very anoxic environments. Stuff's going to suffocate. It's not going to live long enough. Same thing happens with salt, although it's for salinity, not anoxia. Salt layers that form through sedimentary deposition will not allow for bioturbation to turn stuff over, because as the basin gets progressively more saline, before salt settles out, anything that was in there prior is going to just die. So that's not going to work. You're not going to get any bioturbation in continental deposits, especially before the evolution of land animals, because there's just nothing here to do it. Same with the Precambrian, before you had the evolution of like a lot of multicellular life. Any sedimentary records from there shouldn't be tossed around by marine organisms. And then this is the one thing that's actually really weird. Turns out there's an entire class of bioturbation. It's called cryptobioturbation. It's not a rescue device. It has been directly observed off the shore faces of Georgia. And what it doesn't just not disturb the sedimentary layering, it actually enhances it. This is from things like bacteria and other things. The paper I'm referencing here is from Pemberton et al. 2008. It's called Biogenic Chaos, Cryptobioturbation, and the Work of Sedimentologically Friendly Organisms. And a quote from the paper is that, as improbable as it initially sounds, sediment can be pervasively, potentially 100% bioturbated, and yet have suffered remarkably little distortion of the original bedding. Owing to its diminutive and obscure nature, cryptobioturbation has not received widespread attention. As the name implies, it's characteristically small in scale and subtle in expression. What sets it apart from other forms of bioturbation, however, is its tendency to preserve rather than destroy the sediment's primary physical structure. So a lot of sequences that look like they don't have any bioturbation have been re-examined, and this is what they're finding instead. They're finding traces that match preserving bioturbation that is observed in modern nearshore environments. So there you go.
Beautiful. Thank you. Zach, what's next?
Let's go to 37.
37.
We will knock this one out really quickly. There are... I like to think about like evolutionary forces as and maybe this is physics envy, but I like to think about evolutionary forces as strong and weak forces. And there are effectively 4 strong forces of evolution, and that's natural selection, genetic drift, mutation, and gene flow. And gene flow, this specific one is talking about. So this is that the prediction is that hybridization between different species would be unusual or rare due to the assumption of deep time separating the species. So this is actually like just not a prediction of evolution, because for one, gene flow is one key strong force in evolution. Second is it's not even like historically accurate, and that we've been studying hybridization like during Darwin's time, we were like hybridizing organisms and studying them, right? So like all the way back to Darwin, we had some conception of hybridization, but specifically integrating hybridization into an evolutionary framework was a key part of the modern synthesis. And I'd like to introduce all of you to a man named George Ledward Stebbins, who was one of the founders of the modern synthesis and who worked specifically in the hybridization component and was writing about the evolutionary consequences of hybridization as early as 1940. So from the very beginning, hybridization has been a key part of evolutionary theory. It introduces novelty. It's like sort of like akin to mutation in this respect, right? That it's bringing in novel genes into a population, and then that population can diversify in ways that it couldn't have before now that it has this new novelty. So from the very beginning in theoretical population genetics and the modern synthesis, hybridization has been a key part of it. Quick and to the point.
Beautiful. Beautiful. Thank you. Jackson, what do we got? What's next? Oh,
17.
and actually, before we go to Jackson, I'm just going to say this will be our last loop. So we'll have each done 4. We'll have covered 24 out of the 40. It will probably take us close to 2 hours to do that, which is a really nice illustration of the BS asymmetry principle at work here.
Actually, in that case, 20. Actually, in that case, 20. I'll have 20 to be my
right,
last one. Yeah.
Priorities. All right. Number 20.
Well, it's hard. It's so hard to pick because there are so many more that we haven't done.
What was 17?
Anyway.
What was 17? Was Sea Creatures Land Animal Not Become? Oh, that's dumb. Yeah, here we go. 20.
I had a funny story for that, but I was like, you know what? No, this is better. Okay. Yes. Okay. So the prediction, according to them, is that evolution predicts that vestigial organs should be entirely useless, and that we have we've redefined the term since we figured out that things like the appendix, which I don't have anymore, thanks appendix, are totally they're actually not vestigial. They have these super important serious functions and everything. We've now redefined it as different or reduced functions. Now, when I say we've redefined it, what I actually mean is Charles Darwin back in 1871 in Descent of Man, because this is the term, or this is how he defined it. He does define a vestigial organ as functionless in Origin of Species, but a whopping 12 years later, he had revised his idea of that and was like, well, okay, maybe it doesn't have to necessarily be completely functionless. Maybe it could be reduced and just changed function. So, for instance, the like femur of whales, they're not using it for walking around on land anymore, like pachycetids were. They're using it for controlling like their penis muscles and things like that. Our appendix is not used for digesting cellulose, or for, let me rephrase, for holding bacteria that digest cellulose, because we don't do the cellulose digestion, because our ancestors were omnivores, despite what some vegans may say. And so ours has been much reduced. In general, carnivores and omnivores have much reduced intestines compared to herbivores as a result of that. And in fact, Phil Center, who's a paleontologist, pointed out that within the creationist paradigm, vestigial organs exist. If you look, for instance, at geckos, there are different geckos and skinks and things that have different levels of reduction in their limbs, or their fingers. So, like, one species of gecko may have 4 fingers, but then its closest cousin has only 3. This is very common among reptiles, and so unless creationists are willing to return to like species fixity of like Linnaeus, they have to accept vestigiality, as in functionless vestigiality within their own paradigm. But of course, we don't. It doesn't have to be functionless.
Stupid Charles Darwin refuting Darwinism. All right. Grayson, hit us.
All right. It is a really difficult decision because there are so many really silly ones.
We could just do a part 2. We could just do another one.
Because like right here, you got one saying that the prediction is no wheels, which is just
Like,
so silly. And then
really,
there's one saying that, no sexual selection for bird feathers, which is hilarious too. But let's go for a little bit more of a serious one. Let's go with prediction number 2 here.
I was hoping somebody was going to do 2. Thank you.
I
2.
just want to
There you
point
go.
out here that in comparison to number 1, if we get both of those on screen here, because they're saying that the failed prediction in the first one was that the cosmic microwave background, the prediction was that it should be lumpy and anisotropic. And now they're saying in number 2 that the prediction is that it should be isotropic. So literally, they cannot even stay consistent in their own criticisms here. But all right, let's talk about the cosmological axis of evil, which is like one of the most hilariously named things. It's a very serious, potentially thing, but very silly name. I say potentially serious because there's not even scientific consensus that it even exists. Like, there's lots of people that say that it's just an artifact of data processing because, okay, this is I don't know how to technically get with this, but you can't do a Fourier transform on a sphere. You have to use like spherical harmonics, and there are different multipodes like for like looking at the spherical harmonics. And the axis of evil is like an axis of anisotropy in the cosmic microwave background that only shows up when you are looking at like the very like large angular scales, right? So there's only like in the octopole and the quadrupole that's the only time it shows up, right? When you look at other angular scales of ways that you could process this data, which means like kind of more finely detailed, you're no longer seeing this axis. So lots of there literally is not a consensus for whether or not this axis of evil like even exists or not. But I just want to point out that like even if it did, like there is nothing inherent to the idea of inflation or the Big Bang that would state that there cannot be an axis to the universe, right? I mean, there are tons of ways from like primary anisotropies from like vacuum fluctuations that are blown up by inflation that would allow for an axis in the CMB, or like rotational models of the universe. There are plenty of other models that still fit within the basic Big Bang inflation paradigm that do accommodate for such an axis. But again, we literally are not even sure if this axis exists or if it's just an artifact of data processing here. So like for them to act like it's like this huge like defeater and nail in the coffin and the Earth has to be the center of the universe now because there's some axis in the CMB that kind of looks like maybe it fits the ecliptic of our solar system, which like when you actually look at it, it's like it's very approximately in line with the ecliptic. Like when you actually look at it like on the quadrupole, you're like, okay, well, it kind of passes through part of it, but then another part of it is like not in line with it. I don't know. I really, it's a mystery for sure. Like the court ruling is still out on whether or not this axis of evil even exists. So, you know, maybe the creationists should just wait a little while longer before they try to act like it's some sort of kryptonite to the Big Bang or inflation. So,
thank you, Jordan.
I'm gonna shock everyone by going to biology, number 23.
Which number?
23. So,
wow.
Joking about being an engineer aside, I probably don't even have a full high school education on biology. I'm an idiot when it comes to that because I was a creationist when I was growing up. Number 23, though, says that independently originating similarity shouldn't exist. So basically, you shouldn't have convergent evolution is their alleged prediction. But crabs exist, therefore, it is checkmate evolution. This is stupid. And even I, as someone who knows barely any biology, can tell you it's stupid. Let me look at it as an engineer, because they always like to take like God's a designer, so let's have engineers do biology or whatever. Look at pyramids, right? All pyramids are roughly the same. They're a wide base with a narrow top. Why? Because they're all solving the same problem. You need to stack bullshit in a pile. That's how you're going to do it. And that's the same whether you're in Russia or Antarctica or wherever, you know, anywhere on Earth, anywhere with a gravity well, that's how you're going to do it. Same thing here. If multiple organisms are like, let's say being a triangle is the best shape. Triangle is the best shape. Well, you can get a triangle by like folding yourself. You can get a triangle by cutting pieces off. You can get a triangle by ripping triangular shaped pieces off other organisms and wearing them like a hat. Like there's all kinds of ways to get to a triangle. And so like just from the fact that organisms on the planet are subjected to similar pressures, so they're going to come up with similar solutions if those are optimal solutions. Like, this is just so stupid, and it requires no biology understanding to know that.
Yeah, I'll just add that I taught invertebrate zoology in graduate school, and Jackson will know this, but we talked the idea of a BA plan, which is like this conception that there are some general animal body plans, and like these convergent body plans that just like work in certain environments. So if you were like a sedentary organism that filter feeds, there are going to be like certain structures that you are just going to have because of the lifestyle that you happen to live in. So like whether you're a barnacle, whether you're a polychaete worm, whether you're a lophophore, like whatever you are, you're going to have some kind of fan-like structure that can filter out of the water, and you're going to be sedentary. You're going to be attached to some substrate, right? Like, and you're going to have some mechanism of doing that. And like that's convergent across many organisms because of the lifestyle that they occupy. So like it is absolutely a prediction of evolutionary theory that we should see these sorts of convergent structures.
And I'll just point out here that Darwin observed this. It was one of the things that made him, because he's very famous for studying the Galapagos Islands, and one of the predictions he actually made was that island things should be closest to their mainland counterparts. And what he found was that morphologically, across different groups of islands in the world, all of the island things all looked like each other. Obviously, they couldn't have come from the same mainland. So, like, again, evolution predicts this thing won't happen. I don't know what this author is talking about, because this is an idea that comes from Darwin. It is consistent with what Darwin observed, and he pointed that out in the 1860s. I don't even know, like, why. I don't get this prediction as a prediction that is supposedly something that evolution says. It doesn't, like, we've now heard from an engineering perspective why it doesn't make sense. We've heard from a genetics adaptation perspective why it doesn't make sense, and from a historical perspective, this number is nonsense. It just doesn't make any sense. I don't get it.
Can I also point out that like there are very, very many contingent evolutionary events and organisms that we're aware of. Gould famously made the example in A Wonderful Life of endosymbiosis. This is like the most contingent thing ever, because it took 2 billion years for this association to occur. There's no other way to write that except as contingent, right?
Right.
And there's a really good paper by Zachary Blount, the guy who did the CIT Plus, in which sorry, Dan, did I beat? Well, he
Very
mentions
exciting.
Welwitschia, which is a plant for which there were no analogs anywhere else, right? The fauna of like New Zealand, you don't find it anywhere else in the world. So when I think also there's a bit of hand overplaying with the contingency thing, because while yes, contingency obviously does happen, I think you also have to realize that convergence happens. Convergence obviously happens. There are also lots of things that are very obviously contingent, and those kind of get sidelined by this argument. And I had a whole conversation with Inspiring Philosophy about this several years ago. So it gets into weird theological angles too.
Well, that one was kind of a double whammy. 23 and 24 are both kind of about convergent evolution in a way. Just one's more.
Oh yeah, right.
Yeah. Maybe if I can. So like this Ernst Mayr quote is like a frequent quote that's taken like grossly out of its context. And so if I could just like maybe quickly give the context for people that may not have this book, because this is not a book you can just like look up the PDF. So it's kind of hard to get the context for this quote. But specifically, so the quote that they're using here is the search for homologous genes is quite futile except in very close relatives. And creationists use this quote all the time to mean that like the tree of life literally shouldn't exist, or that like evolution didn't predict that there would be homologous genes at all. And that's not at all what this quote is about. This quote is specifically about when you see parallelism and convergence. And so he's talking about when you see two organisms that are incredibly distantly related, that have the same, like very similar structures, like wings in an insect and wings in a bat, for example, that we shouldn't look for a wing gene. that more likely than not, they came to those genes they came to those structures in different ways. So that's the context of this particular quote, not about phylogeny or about whether homologous genes exist at all, but about whether or not we should expect to find homologous genes when we see convergence.
I put that down as 23 and 24. We knocked off both of them there because they just go right into each other. That's perfect. So I'm going to bring mine down to, we're going to go down here to number 38, because this brushes up against something near and dear to my heart, mitochondrial coalescence and all the nonsense that creationists do associated with that. So there's a few things going on here with number 38. I have to set my clock for myself so I don't go too long. Because, so there's kind of two ideas that are thrown together here. The first is from the, I'm sure Zach or Jackson could remind me of the authors of that paper, the Why Should Mitochondria Define a Species paper, that create, yeah, that's the face I make too, because this is
a terrible paper. Like to be clear, it's a terrible paper published in a garbage journal. I just want to be clear on that.
Dang,
Yeah.
Zach, tell us how
And
you
they had to write like a what do you call it? They had to put like a hey, read this at the beginning of the paper to be like, we're not supporting creationism with this.
Yeah.
We just want to be clear.
because
This journal doesn't even have an impact factor. Like, we're literally talking about a garbage journal. Like, the only reason, sorry, go ahead.
So this paper was basically showing that let's put aside that it's a terrible paper and a terrible journal, put all that aside. What creationists pluck out of this paper is it shows using mitochondria as the basis for looking at the most recent common ancestor for species, not the rest of the genome, just mitochondria. You find that most extant, I forget if it was animals or vertebrates. I forget the extent of the sampling. Somebody can remind me if you remember, but it was of a bunch of species.
They look
Yeah.
similar.
And they have their coalescence in a kind of relatively narrow in geological time point in history in the past. And what creationists do, even though it's like hundreds of thousands of years is the range in the paper, what creationists do is they say, see, everybody had a bottleneck at the same time. That must have been the flood, right? That's what creationists do with this paper, even though the way that they calculate where that coalescent point is puts it, in the hundreds of thousands of years at the earliest. And the range is hundreds of thousands of years. It's not that everybody all at the same time had the same population bottleneck. The other thing, so that's the first part of this kind of when they talk about barcoding, that's what that's kind of alluding to. But the other thing here, talking about the consternation expressed by evolutionary authors about basically showing mitochondrial mutation rates, if you assume a constant mutation rate over time, and by mutation rate, I'm using it in the way that creationists like Dr. Nathaniel Jeanson use it to mean mutation accumulation rate over time, you get a recent coalescence for things like the human mitochondrial DNA, where it coalesces within the last 10,000 years. You can't do that with a per generation mutation rate because selection and drift exist. So you have a slower long-term substitution rate. That's the rate at which mutations accumulate in a population is slower than the rate at which they occur. And when you measure that substitution rate over timeframes measured in centuries, and then use that rate to work backwards in time, you get the standard 60,000 at the earliest, 200,000 on the outside coalescent point for your mitochondrial most recent common ancestor for humans. So this 38 kind of actually conflates two things here, that stupid barcoding study, and they kind of smush that together with Jeanson's mitochondrial coalescent nonsense, which is the same nonsense he does with Y chromosomes. But here's one more thing, and I think if I had to guess what Zach was going to go for with this, it's this. Here's one more problem with this. Mitochondria are, because they are only transmitted through the maternal line, have a smaller effective population size than the rest of the genome for whatever animal species you're looking at, which means they are going to be subject to more genetic drift. You are going to lose lineages from your population just by chance more frequently. So you're going to have an earlier coalescent date for the, or I should say a more recent coalescent date for those parts of your genome compared to, for example, everything in your nuclear genome, right? All of your autosomes, your recombining things where you're diploid. You have a larger effective population size, you coalesce further in the past. So even if you take the creationist math at face value for this, for the mitochondria, as soon as you apply it to the autosomes in the nucleus, it falls apart because now you're looking at coalescent dates that are way outside 10,000 years, even when you use the wrong creationist math.
Yeah, the mitochondria is like, for one, it's non-recombining. It's basically entirely functional. It's very gene rich. And so it's like you've got background selection, you've got purifying selection, you've got all of these other things happening on the mitochondria to reduce the effective size, such that and this is what really pisses me off about this paper, is their conclusion is that, oh, everybody went through a bottleneck. That's just absurd.
No.
Just because you see a coalescent ancestor does not mean that population went through a bottleneck. They did nothing in that paper to show, they did no demographic analysis. And we have really good, robust coalescent analysis for inferring when populations went through bottlenecks. They did none of that. They literally just looked at how much diversity there was, said that this would they did some very simple mutation drift equilibrium math and said, oh, it looks like the populations within a few 100,000 year range all have a coalescent ancestor. Well, no shit. The mitochondria already has a low effective size. And so and it has to coalesce at some point. And so it's not very surprising that you would see a coalescence within a certain range, completely irrespective of there being any bottlenecks. It could have been a constant population size the entire time, and you would still have found this result. It's a garbage paper.
My thoughts exactly. All right, so we have reached the end of four runs through the panel. We have covered, because we kind of doubled up there just a minute ago on 23 and 24, we've covered 25 out of the 40. So what I want to do, we've been going for almost 2 hours. What I want to do is, can we do one more run through the panel really quick, 1 minute per person? You got any that are super duper quick? Can we do it? Mr. Wilford, can you do it? I'll come back to you, Zach.
Yeah. Pass.
Okay, hold on. Let me
super quick one.
I've got one.
Hit me, Jordan. Hit me with a super quick one.
Number 6.
Number
there
6.
would be no magnetic field.
I think, did we already do number 6? No, we didn't do number 6 yet.
Field.
This is the stupid magnetic field one.
The prediction they have is that there be no magnetic fields on planets. Planets have magnetic fields. This is talking about decaying magnetic fields. Basically, if you click on the thing, they're saying that magnetic fields are decaying at a exponential rate. That's just wrong. They're not. It's a lie. So
there you go. Done.
Okay, I got one next.
Grayson.
We can go reverse order.
Okay, cool.
Okay, number 35.
Number 35.
This one is just hilarious to me, where they talk about the prediction of bird plumage.
That's what I was gonna do. I'll find another one.
Okay, this one
go.
This was just so freaking funny to me, because they have this source right here where they say the experiment showed that peacocks with a tail, the prime example of sexual selection, are not preferred by females over males with their tail cut off. And literally, the source that they cite for this literally says that the peacock's tails are necessary for peacock mating. They absolutely did not cut any tails off. They literally, their source contradicts what they're trying to attribute to their source. And for some reason, this point is saying that sexual selection is not real. That's the most absurd, preposterous thing I've ever heard.
Ridiculous. Okay, it sounds like they were lying again.
Shocker.
Jackson, Zach, or Mr. Wilford, what do you got?
36.
36. We'll just go right here. Oh, good one.
They're just lying. Again, the Damaralan mole rat has the exact same social structure as the naked mole rat. They're just lying. Sorry.
Well,
Next.
that was easy.
All right.
Knock that one off real quick. All right, Zach, what do you got for us?
I'm going to do 34.
We're going to go up to 34. All
I'm
right.
going to do 34. And this one's kind of it's a little serious, but I'll try not to get too serious. This is the prediction is that under Dawkins' evolution-inspired selfish gene hypothesis, step parents should be less devoted to child rearing than biological parents. And don't get me started on biological determinism, but the key thing here is that parenting and human social structures and culture and things that we do in society aren't a very tiny proportion is determined by your genes. The vast majority of the variance in things in behaviors and cultures, etc., are environmental. They are not genetic. And so this prediction would only be a prediction if parenting was entirely genetically controlled. It is not. That is absurd. I'm done.
Also, can I just point out that this gentleman does not seem to know how IVF works? That's in vitro fertilization, for those who do not know. That is a fertility treatment for people who either cannot have children through non-medical interventions, or for people where both parents are carriers of a recessive genetic condition, and you need to be able to test the embryos so your kid's not homozygous recessive for that condition. Ask me how I know that. In those situations, the children are the biological children of the parents, and the author does not seem to know that that is the case. So,
also, I just want to point out the author's really, really obsessed with Richard Dawkins.
Very.
It's the only evolutionary biologist they actually know.
That's the only one. Okay, that was number 30. We're going so fast, I have to remember to write these down. That was number 34. Okay, Mr. Wilford, you got one for us?
Number 17,
Number
saying that
17.
sea creatures and land animals would not commonly be found fossilized together.
Oh, good one.
Estuaries exist, for one. Nearshore environments often will also get things that get like washed out to sea. But on top of that, there was an ICR article that I read like back in 2018, I think. And one of their writers had been arguing that there was a big formation in, I think it was South America, that had just a big jumble of land and sea creatures together. And when you actually read the papers, the stratigraphy of that formation, it was like stacked on top of each other, a floodplain layer, a marine layer, a floodplain layer, a marine layer. You never saw terrestrial animals in the actual marine horizons. There was very clear organization within the formation. It wasn't a jumbled mess, which just means that formation was deposited on a coastline that had lots of rises and falls, probably due to something with glacial eustatics. Not really surprising.
There you go. All right, for my final pick, I'm going to take us to number 27, because this one's kind of dumb, because this is an example of what I talked about at the beginning, where we just didn't have full information about things. So this one is all about how we thought gene control was relatively simple and linear, kind of a one-dimensional thing, where you have control elements immediately followed by genes, and then different proteins interact with control elements. That affects the expression of the genes. You know why we thought that? Because the first thing we figured out was the lac operon in bacteria. So we thought, oh, that's how genes work. Why don't we look at other things and see if that's the case? We think these are how they work. Let's see if it actually is. Turns out eukaryotes are, shocker, more complicated than prokaryotes. Incredible, right? So that's kind of the big picture thing here, that basically we thought that genes operated and gene regulation operates a certain way because the first thing we figured out in terms of gene regulation is prokaryotes. Turns out eukaryotes are more complicated. I have a question for the author, if I could ask about this one. What does that have to do with evolutionary theory? Like, why is that? I don't understand the logical connection here. Like the thing that he's saying as a gotcha, if you step back from the actual content here, is surprise, eukaryotes are more complicated than prokaryotes. Gotcha, evolutionists. Like, okay, and
26 is the same.
Similar. Yeah, I think 31. Yeah, also kind of like that. It's just like, what? What do you mean?
Yeah, so like 26 and 27 are, I guess maybe 31 as well, are like not actually about evolution.
And
they're about they're about molecular biology, right? It's so like the one gene, one enzyme, or one protein, one enzyme hypothesis. That's like a molecular biology idea from the early 40s that like was known to be an oversimplification by like literally 1950. Just a few years after it was proposed, we were like, well, yeah, that's probably an oversimplification.
I literally, that's such like a basic idea. The one gene, one enzyme thing being an over, that's such a basic idea. I cover the progression of that idea in my class when I talk about gene expression and how the notion of it, our understanding of it, has evolved since we started figuring out things like how cells work. Shocker, we learned stuff. So many, and this is like a common theme, and this is probably a good place to wrap up because we just kind of hit three there because they all kind of thematically are the same, where it's like, you learned more stuff. Looks like you were wrong, evolutionists. Like, well,
No, you should
someone-
dogmatically stick to your opinions like a real a real
Look,
scientist.
you should stick to whatever you thought was right in 1960.
Similar.
Hey, the
to
Bible
that,
has never changed. It is always the same.
the exact same thing is true of number 40, which I just wanted to bring up because it's the very
We're
last
never
one.
going to end.
We didn't touch on it, and that's literally just would make my brain happy to conclude on number 50. It's
there
just
you go, yeah.
epigenetics. It's the same thing
yeah.
that you just described.
All right, I'm going to add 40 to that one because, again, yeah, wow, genetics is more complicated than we thought. Amazing.
Which
Yeah.
Ernst Mayr wrote about in 63, by the way.
Yeah. So
And the creationists are trying to act like Lamarckism is making a comeback now.
that's my literally my undergrads that I'm teaching right now could explain why epigenetics is not Lamarckism. We literally cover that in an intro bio class. Like my undergraduates can explain this better than your average young earth creationist, which should not be a surprise to anybody. But
Quick,
like,
end the stream before 2 hours.
all right, so we have arrived at our 2-hour mark, and we've managed to go through 5 times with some bonuses. So out of the 40 we covered, I stopped keeping track of my tick marks at some point, but we covered close to 30 out of the 40 there. And I hope everybody who was watching saw thematically some of what was going on here. A bunch of these were just misrepresentations of a specific paper. A bunch of them were just straight up lies. Just like author said this thing, it was actually this other thing, just straight up lies. A bunch of these were, here's a creationist thing from Answers Research Journal or Creation, whatever quarterly that we like. Let's throw that in there. And a bunch of these were like just quotes from like not somebody doing science, but just like thinking about stuff. And then like, this guy thought something in the 1940s. Gotcha. But the uniting thing here is that none of these actually work. These are all terrible. So we didn't get through all 40, but we got through enough of them. And I hope everyone also comes away with an appreciation of the BS asymmetry principle, because it sure took a heck of a lot longer to go through the 30 or so that we hit than it did to just read them, right? You can put the nonsense out there way faster than you can. So with that, I want to say 2 more things. One is to thank everybody for watching and joining us on this journey through nonsense. I hope you found it enjoyable, but also remember that this young earth creationist stuff that we deal with on this channel is actually deadly serious, because it's all part of the ongoing Christian nationalist project in the United States. So that's why we have an obligation to push back on that in every way, and that includes their stupid science stuff. Also, if you enjoyed your time here, please hit like. I see 79 people here. That's pretty awesome. And so hit that like button if you wouldn't mind. I see 65 likes. That's a pretty good ratio. So if you haven't already, hit like. And the last thing I will do is thank my panel and just remind everybody where you can find everybody. So Mr. Wilford has a wonderful blog that teaches me geology. It is not linked down below because he was a late addition to the show, but I will add that. Zach has his channel linked down below. It's all genetics and evolution, education and creation debunking. Jackson, also linked down below, is all about evolution and biodiversity and also creation debunking. And it's I recommend that Ancestors Tale series that Jackson is doing. It is a fantastic trek through biodiversity. Highly recommended. Jackson has also written a book co-authored with our friend RJ Downard. Jackson, do you want to pitch your book real fast so everybody can get your book?
The Rocks Were There, Volume 1, which is a play on Ken Ham's Were You There? We're working
Thank
on
you,
Volume
everybody. Everybody should check out that book. It's a fantastic anti-creationist resource. Grayson has his channel, Base Theory, and is a common face on all sorts of debate places, pushing back against pseudoscience all over the YouTubes. And Jordan is the co-host on the wonderful Reason to Doubt show. And I neglected to say that Grayson is linked down below, as is Reason to Doubt. And everybody should tune in to Reason to Doubt for your pseudoscience and other debunkings. And it's not always like it's also like things that you wouldn't expect. We're not just making fun of creationists here. It's kind of look at Jordan. I'm trying to explain. It's like
we're a
bad ways of thinking, right?
We're a
It's-
skepticism
yes.
channel. We're not an
Thank you.
atheism channel, though we are both atheists. So we talk
Thank
a lot
you.
about religion, but we are trying to promote skepticism, and you can be religious and still skeptical.
There you go. And of course, I am Dan at Creation Myths. You know that because you're here watching this. If you haven't subscribed, please subscribe. I do creation debunking things, mostly in the genetics realm. That brings us to the end of our show. I appreciate everybody watching. Thank you. And remember, anytime you're dealing with the creationist nonsense, don't get fooled. Have a great night, everyone. And now I have to find the end the show button, which is over here. Good night, everybody.